So this is entirely new space for me. I've never worked on MED13. We've been kicking around this idea for about a year now. It was literally just born out of Chad Gruder is a knock away from me through the wall. We are side-by-side offices, and sometimes we'll just be talking science, and he was talking MED13 one day, mentioning that it might be good to boost its expression. I go, okay, I'm pretty good with genes and thinking about RNA regulation. So we sat at my computer for about 15 minutes, and this idea was born. And then it took a little while to get the funding rolling through, but we finally got funding through in March, I believe. So it's early work, a lot of conceptual here, and we do have some small bit of data on just some of the intro studies. But so MED13L is obviously a target for gene therapy. We're increasing MED13L expression could provide therapeutic benefit. If you think about it as a haploinsufficiency and a loss of function disorder, there is some discussion about possible gain of function aspects, so this currently would be addressing loss of function. The when, where, and how much to elevate MED13 remains a lot to be determined on that. Questions like, could treatment after diagnosis actually reverse some symptoms? What tissues and cell types must be targeted? How much is needed, and could too much MED13 be detrimental? So gene therapy could involve exogenous supply of MED13 transgenes. MED13 is a very large gene. You'll probably hear me say that multiple times. Delivered by various means. Endogenous MED13 genes, alternatively to classic delivery of exogenous MED13, we could manipulate endogenous MED13 to boost its expression. So while MED13 is new space to me, RNAi-based gene therapeutics are not. I did all my training as a graduate student developing RNAi therapies for a single gene disorder known as Huntington's disease. That's a disease where we actually wanted to turn it off. We were super successful in getting some foundational data. Those therapies are now being tested in the clinic. So this is, again, you've seen this before. I feel like we always need to reiterate, though. You'll have you need like a little certificate at the end of this that you know the central dogma of biology, right? DNA is transcribed to mRNA, which is then translated to produce protein. What wasn't yet mentioned, which always fascinates me, and I tell all junior trainees coming to my lab, is the genome is 3 billion A, C's, T, and G. It's crazy. Like, to me, it's just fascinating that that encodes all of life. And it's a great time to be a scientist right now because we have the sequence at our hands. So my research focuses a lot at the RNA level, where historically we were interested in turning off the RNA of Huntington's to block the production of a toxic protein. We have since been interested in many other aspects of RNA regulation, studying small non-coding RNAs that arise from our own genomes or a host of RNA binding proteins that can regulate various genes. And so in this case, while something may endogenously be putting the brakes on MED13, we think about, can we alleviate those brakes? And we can use definitely RNA-based therapies, are real and already in clinical trials and probably approved. Okay, so what I'm going to talk about today is an upstream open reading frame. So how translation starts, this is a schematic of an RNA where you have some non-translated, non-protein coding region at the 5' end, it's called, so on the left, and the coding sequence in the middle, and then a 3' non-coding region. And so a ribosome is the reader. It hops on this thing and it starts reading it. And it has to find an ATG to start making protein. But while there is a primary ATG at the start of the coding sequence, which is shown here, there's also the potential for upstream ATGs, which make upstream open reading frames and then can be translated. So the ribosome has a job to figure out which one of these ATGs to pick, and it's a really interesting biology there. But what's known is that alternative ATGs or UORFs can basically hog the translational machinery and say, make me, make me. Meanwhile, in this case, MED13L may be at the expense, and you're not making as much of that MED13L as you could. So while this could serve as a regulatory purpose to control when and where proteins are made in cells, and I believe that's entirely true, it's also possible we could exploit this for therapeutic benefit. And so this has been mentioned. Matt briefly mentioned the idea of using ASOs. So this is antisense technologies. If we know the sequence of everything, we can simply design sequence-targeted therapies to target very specific sequences. Once you get to about a string of 20 letters, you're at about a one-time occurrence out of that 3 billion letters based on 4 to the 20th power. So it can have very high specificity once you get these at longer lengths. And so ASOs are already currently being used in the clinic. The idea here is, can we drop an antisense oligonucleotide onto one of the upstream ATGs to tell the cell to get down there to the MED13 ATG? This has been done in some publications, so we think it is possible, which is why we like the idea. There's this paper that was published. When was this? Back in 2016. But as in the case of science, there's always controversy. There's always controversy even in my own lab. We'll find one result one day, and then we're so excited, and the next day we're really bummed. It happens all the time. It's a grind. So there was a recent study that sort of refuted some of these findings. So it's still up in the air. I don't think it's enough controversy to pump the brakes at all. I say we go for it. It's a simple test, and that's why we're pushing forward. I'm sort of like a genome junkie. Much of our major discoveries that have come out of my lab lately have all arisen out of this genome browser. So in this era, you can like hop online, like fly through the genome and navigate to your genes of interest. And obviously, this is a picture of MED13L. You saw it in different schematics showing exons and introns. This thing is covering 300,000 letters, so 320K. It's massive. But we, at the mRNA level, only care about those exons. So this is a stitched view. Once those introns are cut out, this is what the mature mRNA looks like that gets shipped to the cytoplasm and encodes for the protein. So this is, you've heard conservation talked about. This is the conservation track on this. You can see the coding region is highly conserved because that makes the protein. But you can also see some super strong, important stuff happening in these regulatory regions that are non-coding RNA. And we think we can target any of these to boost expression. So where we started was the UORF, because I had another project going on in my lab at the time. I just want to see my timer. Sorry.
5 minutes.
Okay. We had another project ongoing in the lab where we were studying a UORF in another gene that was very interesting. And so when Chad and I were talking about this, I go, well, I'm working on microRNAs. Those are inhibitors. Let's look at the tracks. We have some unique data on that. Nothing super much jumped out there, but I said, hey, let's look at the upstream area of this and see if there's a UORF, because we're working on that. And sure enough, to the trained eye, very quickly, I was like, ooh, we got something here. So this is looking at the 5' end of MED13. This is the canonical start codon to drive MED13 expression. Green is that ATG start codon. If you start scanning back, even you can maybe spot it. Once you get to right here, we have an upstream ATG. Could that be important? We can look at this conservation track. This whole region is kind of important. So there's some regulatory control in here. But this ATG appears to be present in virtually every species. So every species has this, mouse has it. So there's a universal break on MED13 expression. My guess is that it probably is super important during development. That would be the hope, right? And then post-development, you can go in and muck with this a little bit and hopefully therapeutically boost MED13. All right, so let's test this in the lab. So we can basically, MED13 is massive. The easier experiments to make some mini gene constructs. So we just isolated the first exon. There's only 2 ATGs at play here. One is the one we want, driving MED13, and one is this upstream ATG. So we can make these mini gene constructs and put a tag on it. This is just a V5 tag that allows us to detect it in the lab very easily. So in one construct, it's wild type. The ATG is intact. In another, we mutated it. So can we break the break? And we can test this in cells very easily. Insert the constructs into cells, isolate the proteins, run the proteins on a gel, and then probe to detect that V5 tag. And sure enough, the wild type constructs you can see are relatively lowly expressed, and that delta UOR for that delta ATG, we now relieve that break, and we boost expression by about 10x. So this is a very clean result. We saw this with our other gene of interest as well. So these UORFs really do a good job of hogging translation in certain settings. We want to test this, this is in a cell culture, which is irrelevant. This regulatory switch, this break, might not be breaking the same way in a brain, in neuron cells. So we want to test that, and we just now have the viruses to put these mini genes into mouse brains, and those injections will happen this week. So Antisense oligos in Iowa City, Coralville, we have a suburb, massive producer of these little oligonucleotides, which is perfect. We can place orders for little oligos, and they show up at our doorstep the next day. They're also very nice to us in helping with design. They have experts in anti-ASO design, and so this is just some of the comments from them. The sequence is targetable, and the off-target profiles look really great. So we currently just recently got in-house 5 antisense oligos that basically just tile across this ATG region. And with this mini gene construct, we're going to see whether addition of these ASOs can give us the same result that we saw with mutation. Can adding the ASO here tell the cell to use the other, to use the canonical native MED13? So that's testing is going to be happening here in the next month. When you're using mini genes, those are super artificial, so we always want to do this with the real gene. Again, MED13 is big. Even after you stitch all together, remove those introns, MED13, the mRNA is still over 6,500 letters. So these can be major headaches to clone. There's new ways to clone things, luckily, and my undergrad student pieced this together in just a few weeks. So we now have a full-length MED13 cDNA clone. I'm not sure if there's one in existence. Everyone in the community is welcome to have it. Just let us know. And so we will now do the same type of testing with mutation on a full-length construct. Just to make sure these things are working, we put them into cells, and now rather than blot for V5, because now this is no longer an artificial thing that's tagged, this is actual MED13 protein, we had to get MED13 antibodies. We tested a panel of at least 3 antibodies, I believe, and we identified one commercially available antibody that the field may already know about or not. That's the catalog number. It definitely works. You can see when we add MED13, it's every other well there. It's clearly increased. So this is nice, clean data. Okay, so the brief summary here, hope to have more exciting data next year, is that increasing MED13 could provide therapeutic benefit. Targeting natural regulatory controls may be a very attractive way because MED13 at 6,500 is actually too big for viral-based strategies, the ones that are pushing to the clinic faster. There's ways to get around that, but there's a lot more interest now in manipulating endogenous genes using RNAs. And so we think if we can, clearly the data show that this UORF is putting a pretty substantial brake on MED13L. And so if we can remove that, maybe we can help. And so I'm not going to get discouraged even if we don't find that, because I already know I've looked at this long enough. We have a very unique data set in the world. I think we're probably the only one who has it on microRNA binding events mapped to this in the brain, and so we have the key that will tell us what other regulatory sites, if this doesn't work, will we go after next. And so we will just continue our work on ASOs, and we'll find some sites, whether it be here or down here, that will boost the expression. And then hopefully those can go to those animal models, and I can just knock on my door and say, Chad, here, put these into your mice. So thank you for your attention.